Thank you very much, and thank you, Natasha, for that intro speak. Wednesday breakfast in Huddinge, Flemingsberg. This is quite of a difference to Solna. Is it this really every Wednesday, Natasha?
Yes.
That's great. We should definitely come here. Now, great to see everyone. I'm Jonas Båtelson. I'm going to talk about CuraCell today. Yes, I've been engaged in some other startup companies in the healthcare field, and also previous history in the MSC field also, kicking off both Jonas Ba and cell collabs in this space. But today we don't have time for that. I'm going to talk only about CuraCell, and I'm going to start up in the presentation to get your perspective a little bit about the journey, because most of the science that we have in biotechs obviously stem from academia, one way or the other, regardless what the pharma company says. And CuraCell is not an exception, but it's a sort of a different story to many else. So the original research comes really from a group of immuno-oncologists here at Karolinska. We're working on a T-cell protocol, and I'm sure that everyone in the room is quite familiar with that since we're at the ATMP cluster. It's a polyclonal T that we're taking out in the first round. So original research here, and the researchers' aim at that time with a collaboration with NCI in the States was really how to grow T cells for really tough cold tumors with few and exhausted T cells. And they ended up collaborating with a group of German clinicians in Frankfurt who were really interested, namely Professor Elke Jäger was heading up the oncology branch at the time in Frankfurt, who was really intrigued by this research. And Elke had the opportunity to, like you have in Germany, to actually work with patient-named treatments. And that's a little bit like swearing in the church when you're in Sweden, and definitely in the US. It's a version, obviously, of hospital exemption and patient-named treatments. And she got actually another local German producer. It was a research institute, now it's a CDMO, to continue the work of the researchers in Karolinska to develop a full-fledged actually protocol for an autologous T-cell product, and to put that directly into patients. So all this was done before we even kicked off CuraCell, and Elke has actually gotten to start to treat patients on, you will see, a set of really, really tough indications in oncology, terminal patients, and the clinical outcomes, still individual, case-by-case results were coming out and showing really interesting data. So where do I come in? Well, I'm more of an entrepreneur. I spent most of my career in management consultancy, working for medtech companies, healthcare, and pharma, of course, also, but not specifically so much actually in pharma innovation before coming into this space 4 years ago. But I added something to the team, and that was the complexity of bringing these products up to large scale. Because what we're working on here is highly complex from an industrial point of view, both in terms of making sure that these products will not cost a fortune in the end, they will not fit into the payer systems. And the other part, integrating them in healthcare, requires quite in-depth healthcare process understanding. So what I saw with this protocol was not only that you can deliver breakthrough science, but also that it's a protocol actually more fit into the payer models. This will not cost a fortune. This is possible to put into more of a large-scale manufacturing facility. And that was the trigger for me to actually bring together a quite spread out group of IPs, individuals into one common company. So, telling you this journey, because every start is not very simple. Here we have a brilliant professor, coming up with a great idea. And here's just a bulletproof blueprint model of how to bring the project forward. So where are we today? Very quickly, so we're located in Solna. We're in clinical stage. We activated the trial last week, the first phase 1/2A. And we're really targeting cold solid tumors. So we're targeting those tumors where few T cells in really harsh tumor microenvironments, where we don't have solutions for terminal patients today. Setting up, we founded the company in 2022, and of course, as with the background that I just mentioned, but choosing really here, Stockholm and Karolinska as our main ground for the R&D center. Although obviously we have a clinical operations team in Germany, because our clinical journey based on the background, obviously start the first formal trial at the same hospital where the patient-named treatments were carried out. I'm coming into the product because at the core of our company is really a T-cell expansion and enrichment platform that drives cytotoxicity significantly, but it's a combination with a very efficient manufacturing process that is really the key to the company. Manufacturing today is, as I said, it's the same producer that was part of the foundation of the company as well. So that's been the easiest setup to quickly come into a clinical proof of concept for us, given the background. Otherwise, of course, we would be at Vecura and of course locally here with all other research activities, which we might be for next project coming up. So yeah, you know about the different modalities in this space. The CAR Ts are much known, and the concept for autologous T-cell therapy is obviously the extraction of T-cells, the expansion, modification, and then through IV infusion, taking it back to the patient to spur obviously the immune response. What we're going to talk more about here is the polyclonal TIL, the very non-selective, and the TCRs targeting the solid tumors, because that's really the space that we are addressing. And looking at the big quest that we are addressing. Oh, now I see that there might be some flips in this PowerPoint deck. Anyway, looking at the unmet need that we are targeting is really the tough cancers and the terminal patients within cancers like prostate, colorectal, breast, glioblastoma, and so forth, obviously. And here we have staggering death numbers. Everyone knows about that. A little simplified, you could say that the CAR Ts really struggle, obviously, to come into solid tumors. The TCRs are very promising, but they're also very narrow. They are targeted therapies, And the challenge for that, we had Adaptimmune being sold, the first TCR on the market in the US. The value that you bring forward is obviously much less than if you can go into broader patient categories. So, and from a market perspective, you could say that you can solve some of these cancers, but it might not really flip the coin when it comes to saving a lot of patients, right? So, interesting, but quite narrow. And TILs, researchers would claim it works on melanoma, maybe a little bit further down the tumor mutation burden register, but not very, very far. They're going after lung cancer, head and neck, and those types of indications, but not really what we're talking about here. And of course, these companies try all sorts of different approaches, but either they are very targeted, as I said. This is a quite bold statement, by the way. They're doomed to become niche markets. It's a sales presentation. Or you can work with a lot of gene modifications, because the polyclonal TILs, our colleagues in business, they of course try all sorts of methods to try to impact the tumor microenvironment in different ways. It's just that it becomes quite expensive to do so. So then we're back to, does this really fit into publicly financed healthcare systems like we have in Europe, for example? Question mark. Well, very quickly, I go into sort of the solution that we have. The expansion and enrichment platform we have with an engineered process can create a T-cell product with a phenotype that is very, very superior when it comes to the memory subsets and also the effector function. And the very short version of it is that we can go into these cold tumors and address a very harsh tumor microenvironment to the extent that we actually can treat these late-stage cold solid tumors. So this is very much the summary of the two major statements. But it wouldn't be interesting to bring this forward if it wasn't for this. So we're bringing forward a process that is very simple, yet not simplistic in its approach. Means that this will cost significantly less than what you see in many other T-cell therapies being approached now to the market. And that is important, because the industry wouldn't be interested otherwise. So I'll give you a few things about the platform very briefly. I mean, we're working on the traditional expansion protocol of T-cells is to expand with interleukin-2 as the growth factor. And already discovered by the researchers here at Karolinska, they were trying out a lot of different interleukin sets, obviously, to try to improve the phenotype and the expansion and proliferation of the cells that work specifically for these cold tumors that they addressed. And IL-15 and IL-21 turned out as very, very interesting. IL-15 to add stemness and IL-21 to add effector function. But that's not really the magic, because you can try this out. There are many research articles showing that yes, you can improve a little bit. It's the engineered process itself that we work with that really improves the phenotype of the product significantly, getting a much higher memory subset share and also effector function of the T cells in our final product. We don't reveal all those juicy details. The only thing that we reveal at present is actually one of the filings we have through the patents, and that is actually the composition of these T cells itself. But there's a number of other factors actually impacting it. And then you can question, why did we come up with this? Well, it's two factors. One is that this research team was really focused on how to expand T cells for really cold tumors specifically. So they tried out probably more expansions than anyone in the field at that time. And then it's the combination of having real patient cases that Lucas has had and really used the bioinformatics of that to do sort of the re-engineering of the product in the development of the final product, I would say. Yes, there's plenty of data, of course, to show the superiority of our expansion protocol. I will not go into the details. I would like to mention, before I come into the clinical data, to show you that the protocol that we're using on is, of course, the cytopad platform brings forward our, you could say, our main product. And that is a quite simple process in itself, highly efficient, that allows to add more clinical complexity. And I think that looking at the field in general, everyone tries to please the pharma industry by doing sort of one-dose treatments and trying to look as much as a drug as possible. Adding repeated dosing is not that complex. It's not that costly, but it definitely has a synergistic effect in treatment. Everyone knows about it. We also add checkpoint inhibition for synergistic effect. That is maybe not so revolutionary. Elke was early on it, but now obviously a lot of people follow. But this is the rationale, because we're really trying to go after really, really cold tumors. But when Elke did this, before we actually kicked off the company itself, she was very exploratory. So I'm just showing you 11 patients that were treated with Cytoply, the product itself. You can't see this as a trial, of course, everyone knows that. These are individual patient cases, but they're highly interesting, every one of them. These are late-stage patients, terminal. They basically have no hope left, right? Ranging from a few months to live to a little bit more, obviously tougher than we would include in the trial. But the results, and also worth mentioning, they were giving either 1 dose, 2 doses, 3 doses, and only one of the patients actually received the combination with checkpoint. And that was the prostate cancer patient who's now a 5-year survival. I think perhaps more interesting is also the glioblastoma patient that we published this spring and presented at the AACR in Chicago, or Lucas did, which received 2 doses and achieved a complete response. So obviously this product is highly potent. The publications of these cases didn't come out until this spring, and the prostate cancer patient and the glioblastoma patient is now published, and the pancreatic we hope to get published fairly soon. So I think, and before that, Elke had clinical outcomes, 21, 22 and so forth. It's not really interesting until you really understand the underlying mode of action and mechanism and how it works, and can build a trust that you can do it again, of course. So I think that publishing these results over the last year has definitely sort of increased the credibility of what we're doing, but also helped us in building up the hypothesis for the clinical trial that comes up. I encourage you to read these papers, and of course there's a lot of data showing things like this where we really can document the PK/PD profiles and the mechanism of the product. So where are we today? Well, we have a first polyclonal TIL application, and we choose polyclonal TIL. Of course, we believe in it given the clinical background that we have, but Cytoply can be plugged into basically any T-cell therapy. And it can improve the phenotype of any T-cell product in CAR-Ts, TCRs, etc. So of course this is an opportunity that we will explore more going further. The indications that we chose, so we are obviously up against companies like Iowans. I'm quite surprised sometimes that how few people who know about the company, but they are the first TIL product on the market approved for melanoma, now being rolled out in the US, and of course they're knocking on the door in Europe and in Canada and many other markets as well, right? They're obviously starting on the hot side of the tumor registry, and they're also followed by KSQ and Obsidian, two other American friends of ours. They're trying the gene-modified version, trying to sort of improve the Iowan story, you could say, in the hot side of the registry. We're kicking off our clinical journey in a very bold way, people would say. But remember, the whole project was aimed at these indications in the first place. So we believe that going here is more likely for us to succeed. But of course, from a competition perspective, it's also, of course, very beneficial. Sorry for some of the slide formats here. It seems to be some kind of... wait. Yes, so last week we kicked off this trial at the home ground where they treated the first patients. So we're running a phase 1/2a. Prostate and colorectal will be our two first indications. Safety, tolerability, of course, is the main objective, but of course we want to see efficacy in this, right? And we hope to be quite quick. It's a single center, but we're collaborating also with Marburg-Gießen and the Frankfurt University Hospital also, so the uptake of the amount of patients here is quite large. So we hope to be able to execute the enrollment in 12 months. And what are we aiming for in this phase? So, and very revealing, I haven't raised a lot of money for this. We raised around 130 million for this, of which 80 million is spent now in this phase. But of course, this is a result of a lot of research already done before we actually formed the company and the team around CuraCell. Our aims are very obvious, right? We want to show clinical proof of concept. I do want to highlight another thing, that I'm talking about CMC Gen 1 here and CMC Gen 2. So the first CMC protocol that was developed by this German producer was something that I judged, and not only myself, but that this is not scalable enough, right? It's a proprietary bioreactor system that we can take into thousands of treatments. The process is a bit slower than what we are developing now in parallel with more the gold standard of T-cell expansion equipment. But starting all over from scratch, sort of doing the process optimal before going into clinic would obviously have taken at least another year before we can get to a clinical proof of concept. So that was an active decision from our side to basically bring the old stuff into clinic. And we believe that was the right strategy, but it's been a lot of work actually on a CMC protocol that we don't believe is really fit for market. And in parallel run this CMC Gen 2 development, obviously in Solna, with our own team that we will plug into, obviously, the follow-up trial. So that's also one aspect, because I think we're in a field, to sum up a little bit, that we're in a field that we need to drive these projects much longer in terms of clinic and clinical proof points than our colleagues working with small molecules and some of the ADCs, because it's a project that you can plug into very much the existing pharma business. For us, it's quite important to show that this actually works and that the journey to market approval is significantly shorter before we can start, real collaborations and make deals and things like that. So this is an example of some of the decisions that we've had to make to drive the project forward. Yeah, I think that's pretty much it. We're 11 people. We're located in Solna. Might move to Huddinge. Seems like a crowded place. A lot of other colleagues here. Some resources obviously in Frankfurt in the first step. Thank you.